The overall goal of this procedure is to identify in vivo protein-protein interactions of membrane proteins through a membrane strep-protein interaction experiment, or membrane spine. This is accomplished by in vivo cross-linking using the reversible cross-linker formaldehyde. The second step is to purify the strep-tagged membrane bait protein together with the cross-linked prey proteins. Next, the cross-link is reversed by boiling. The final step is the separation of the membrane bait protein and the co-purified prey proteins by sodium dodecyl sulfate polyacrylamide gel electrophoresis, or SDS-PAGE. Ultimately, immunoblotting and mass spectrometry analysis is used to identify the prey proteins, allowing analysis of membrane protein interactions.
I'm Sabine Hunke, and we will show you today the membrane spine technology. The main advantage of this technology over existing methods like co-IP is that it allows you even to detect low affinity or transient protein-protein interactions. Karolin Tschauner will demonstrate you the procedure. She's one of my PhD students who developed the protocol.
To begin this procedure, grow bacterial cells expressing the membrane bait protein with a C-terminal strep tag fusion. Induce the expression of membrane bait proteins at early log phase by addition of 0.5 millimolar IPTG to 500 milliliters of medium. To allow sufficient expression, incubate the bacteria until the late log phase. Prepare to perform formaldehyde cross-linking under a safety fume hood due to the toxicity of the formaldehyde.